I wanted to talk a little bit about how to advance mRNA cancer vaccine technology toward meaningful clinical outcomes from in terms of tumoral heterogeneity, routes of administration, and the development of methods to assess the efficacy processes. Um, that'll all need to be addressed. And I was wondering if you could hopefully talk us through perhaps some of the existing challenges in the short term, what they are, and how, as a sector, we're looking to overcome them. Perhaps we can start with tumoral heterogeneity, moving on to routes of administration.
Yeah, I think one of the important things to remember, especially in the solid tumor situation, is each primary tumor and its associated mets can be quite different. So even within a patient, you can have tumor heterogeneity, and then we have patient to patient. So it's quite broad, this question, and we don't necessarily have the best, understanding totally on this. This is a very rich area of research, looking at microsatellite stability and so on and so forth. I think the other thing to, when we're talking about vaccines to remember is, or to reflect on, is the CAR T cell space has spent quite a bit of time in solid tumors. I was involved with generating a mesothelin targeting TRuC T cell that's now adapted when I was at TCR squared, and it works quite well, actually. Patients that got it had positive outcomes. They weren't CRs, though, and that's what the market is looking for. And in glioblastoma, there's been quite a number of studies where CAR T cells have been intrathecally delivered right into the CSF of these patients, and they're still not getting the outcomes that they want, right? And I think when you reflect on that, and then you think about the vaccines, there's more to this than just activating T cells. And I think one thing that's really important about the cancer vaccines in the adjuvant setting is they show that, within these patients, myeloid cells can take up, in this case, an RNA vaccine. They can process antigen and activate T cells. I think this is step one for us. Now, step 2, 3, and 4 is, how do we make, in patients that aren't adjuvant, haven't had surgically removed these tumors, how do we make then the tumor microenvironment more amenable to being, attacked by the immune system? And so one thing about the vaccines that I think is very interesting, and what Andy at Genentech and the folks at Moderna are showing, is that each one of these patients has a personalized vaccine, which means the antigens within those tumors can be identified, and in not all patients, but a good, I'd say 50 to 60% of those that were treated showed 1 or 2 T cells being activated. So we know the T cells within these patients can be activated, and we know that you can approach tumor heterogeneity in the context of MHC and neoantigen by making these personalized vaccines. So what I truly believe is the next step is to take advantage of myeloid cells that penetrate into these tumors. People often find it amazing when I tell them some of these statistics. Glioblastoma can be 75% blood-derived myeloid cells, ovarian cancer up to 50%, triple-negative breast about 50%, and so on. And these are the cells that often cause pathology, like in a COVID inflamed lung, for example. They're exactly the same progenitor cells, just that the cancer shuts them down. So our ability to activate these cells within tumors, these are the orchestrator of the immune responses. These are the cells that these vaccines are using in the periphery to activate T cells. Imagine if we can get that foot soldier turned on in the TME, recognizing the tumor, killing tumor cells, and then liberating the antigens that today Genentech and Moderna are actually going and looking at ex vivo and putting back in. So to me, the way to approach tumor heterogeneity is really to take that next step and do what is being done ex vivo directly inside the tumor, and it will kill 2 birds with 1 stone. One, it will liberate the neoantigen, and then the second one is it's going to soften up that tumor microenvironment to make it amenable to T cell penetration and killing of the tumor cells.
Okay, thank you very much for that. I appreciate it. I want to, earlier we talked about LNPs, and I want to make sure that we cover that. Like I said, I have the great opportunity of taking some information before the event, and there were quite a few questions submitted around lipid nanoparticle challenges. And so the to sum them up, here is what we're talking about, that many of the LNPs currently available are not biodegradable, and we don't know their interactions with mRNAs. And so what I'm looking to better understand is, since many companies are kind of still flying in the dark in this area and maybe perhaps rushing to solve the problem without maybe the best science behind it yet, can you perhaps provide some color commentary on this statement? Do you agree? Is that your experience? And if so, or if not, just the why behind it.
I mean, point at me. I think, not to sound like the FDA, but each product is different, and each product has its pros and cons, and each product has to be, looked through that lens. So, taking the experience at Myeloid, we've delivered to a cohort of patients now in vivo LNP mRNA CARs. And I can speak to what we see in non-human primates, and I can report that, the LNP partner we have at Acuitas and the library that we use is actually somewhat biodegradable relative to other LNPs. And there certainly are there are ionizable lipids that aren't. And the ionizable lipid, there's some work done, I think, from the folks at Genentech and some others earlier this year that was looking at the importance of ionizable lipid plus RNA. And the RNA alone isn't that immunogenic. The LNP alone isn't that immunogenic. When you put these two things together, this seems to be an important element for driving immunogenicity. But I think if we think about LNPs, I do believe that the Genentech vaccines delivered IV. We deliver our product IV. And so we're doing that obviously to hit as many myeloid cells as possible. I think Moderna delivers their vaccine intramuscularly or subcutaneously. And so different people are trying to modify and optimize based off what they see. But this is actually speaks to, I think there's two big elements here. We're seeing a very strong, patent, infringement and legal situation going on out there around some of these lipid nanoparticles. And I think that's because I'm not so convinced that there is this broad range of ionizable lipids that are going to be amenable to be used. I think when we go through all of the screening and really appreciate this, there isn't going to be the library that we hope exists may not actually be as broad. And so we have to keep that in mind. And then on the delivery side, I think we need to be thinking beyond lipid nanoparticles. I think the future probably is beyond lipid nanoparticles, whether that's a combination of antibody-fused RNA or, biodegradable polymers like PLGA, PBAE. I think this is actually the path. And frankly, having worked in biodegradable polymers in the past, they're actually much more, they're easier to work with than LNPs, but they have a number of pitfalls with being combined with RNA. So, I'm not trying to give a wishy-washy response. I think each product is unique. Each RNA is unique. We have certainly learned through our experiences that you can't just take one construct and put it into the same backbone of an RNA, for example, mRNA, and expect the same outcome. Everything is quite different. And when you really want to sit down and draw out what happens with RNA when you deliver it, you could draw a flow chart that goes out the door because there's so many things that happen. It gets injected, proteins gets to a cell, gets into the cytoplasm, what interacts with the cytoplasm, how does it get to the ribosome, so on and so forth. So I apologize I haven't given you the perfect answer there. I just think it's really nuanced, and there probably isn't one. It probably does speak to the amount of science that I think we're still delving into.
Sure, absolutely. And that's a certainly fair answer. And I want to move on to manufacturing and certainly supply chain. Before we do, I want to please draw your attention to the second poll question that's been posted. Please do respond, and we'll get to our findings here in just a second. But like I said, we want to move on to manufacturing. So mRNA vaccines, they're precise, they're safe, they're flexible. We know this. And they can be easily manufactured on a large scale for clinical grade applications. But, to achieve this, the development of sustainable, cost-effective, manufacturing processes must be addressed, just like any other component of the cell and gene therapy space. And so, Andy, I'd like to first talk to you I want to talk a little bit about the availability of raw material and just how much of a challenge that actually is, from your perspective.
Yeah, sure. So the raw material availability and the COGS question kind of go hand in hand, because one of the things that's sort of a key factor for these types of therapies, not just RNA technology, but the CAR Ts, stem cell technologies, gene therapies, is that the cost of the materials is disproportionately large compared to other pharma products, partly because of the scale component and partly because there's just not that many manufacturers. It's still a very immature industry. Manufacturing the key kind of materials that make up the components of these therapies is still very immature. It's very difficult, and there's not that much competition. And so you have the challenge, and one of the things that I focus a lot on at Genentech is you have the challenges of, okay, if this thing works, we've got to figure out how to scale it. And to scale it, we need to make more of it. And we also need to raise, to a GMP level quality. Most of the stuff in the clinical space is not GMP quality. And so all of those are fairly, non-trivial challenges to be able to actually commercialize one of these therapies. And the raw material sort of, if you look at the BOM to make something like this, it's hundreds of products, but there's a relatively small number of those products that make up a very large portion of the overall COGS. And so obviously we focus on those items and work with suppliers to improve quality, improve yields, improve efficiency. And, it's a journey that we have to follow to be able to commercialize a therapy like this.
Sure, sure. And just as I suspected, the poll results are they're live, and just as I suspected that the largest percentage of our listeners today listed that raw material quality is in fact their biggest challenge. And so I suspect, certainly based on everything you just said, that you're in agreement, understanding. True.
Yeah, and I would extend that one step, that the raw material quality at scale is really going to be the challenge. And it's two separate elements to the same challenge. So we need to make more of it at a much higher quality.
Sure. Yeah, fair. Daniel, I wanted to check in with you to see what are, in terms of just mRNA cancer vaccine manufacturing overall, what would you say are the biggest bottlenecks we're facing in manufacturing? And what are the most cost-effective, steps perhaps we can take into 2024 to address that?
So I'm going to give the kind of small biotech startup answer to this, which is going to be different for others. I think access to technologies like capping technologies and LNPs are a bottleneck. It's expensive to use proven LNP technology and capping technologies. If you want to make the best product, you could see yourself in a situation where you need to combine or stack royalties. So there's kind of like this balancing act that needs to happen across there, which granted is less of an issue as you expand and you look at larger companies. So I think that's one element. Capability and people that understand and appreciate the space. There are components of RNA that are very simple in bucket chemistry. There are components that are very, very unique and that are very different to other things. So you can't just take a biologics expert and expect them to just pick up RNA, which I think sometimes is something that is done there. And then like always, when you have a starting industry, access to CDMOs and folks that know what they're doing and can consistently, continuously produce quality product is another element to it. I think if you were to start today and say, I want to make a GMP product, and you had even if you had the resources, you're probably still looking at a 6 month, maybe even a year delay by the time you can get into someone's, pDNA, depending on how you generate your product, pDNA, linear template, so on and so forth. So I think they're the challenges that, at least from my perspective, that we encounter, at our scale of business, not necessarily scale of what we make, but scale of business.
Yeah, sure. That makes sense. And Andy, what's your perspective from, the kind of the opposite end of that bell curve, from Genentech Roche's perspective? What are sort of the biggest bottlenecks facing mRNA manufacturing today?
Yeah, so it's not actually that different from what Daniel said. You do have to look at the technologies and the implications of those technologies in terms of royalties, again, when you go to scale. And I think if I look at, if we move to the next, like currently we're manufacturing in going into 2024, we'll be manufacturing individualized vaccines to the level of, say, high hundreds of patients, less than 1,000 over the next 18 months or so. To take a commercial strategic roadmap to a commercial launch for a therapy like this, you have to go to sort of the high thousands, right? Over 5,000. And so at least one order of magnitude greater than what we're doing now in the clinical phase. And so that means all of the key components that go into the manufacturing of this has to be scaled by an order of magnitude. And the industry is not ready for that yet. So plasmids is a big challenge. Getting the plasmids in a very short time period that are custom, synthesized is a big challenge. Getting them at GMP grade is a big challenge. All of those are things that, and many other materials like that are sort of the key components of this technology are things that have to be much further developed to be able to assemble them into a manufacturing facility to actually manufacture at scale. And yeah, so there's a lot of people in the industry focusing on how to do that. And that's a key element that we have to deliver on, or we won't have a product, basically.